Stem cells are powerful entities within our bodies that allow us to regenerate and replenish damaged tissue so we can maintain our consistent functions on a daily basis. Skin, gut, and blood cell regeneration can all be traced back to stem cells. However, not all tissues in the body have stem cells, such as the heart. They cannot be regenerated during adulthood. Scientists discovered a method to establish pluripotent stem cells from pre-implanted embryos that were donated from patients from in vitro fertilization clinics. These specific pluripotent stem cells are called embryonic stem cells. With the right conditions and environment, pluripotent embryonic stem cells can self-renew unlimitedly in a culture dish and develop into all types of cells in the body, holding the highest potential for clinical and therapeutic applications. For example, a severe burn victim can receive an organically grown skin graft developed from embryonic stem cells without having to use the patient's own skin from undamaged areas for grafting. This means there is an unlimited amount of skin cells to be given to the patient, thus being treated more effectively. One issue with this method is the cells generated from embryonic stem cells do not possess the same human leukocyte antigens as those of patient recipients, and thus, when embryonic stem cells-derived donor skin cells are applied to the recipients, the immune system will recognize them as foreign objects or threats and attempt to destroy them. The way of solving this challenge utilizes a technique called somatic cell nuclear transfer. Somatic cell nuclear transfer involves injecting an enucleated egg with a nucleus from the recipient's own somatic cells, for instance, a nucleus from a skin cell or a blood cell. The eggs with recipient nucleus further develops in a cultured dish to an embryo that is called a blastocyst. A blastocyst is a pre-implantation embryo, 5 to 7 days post-oocyte fertilization in humans, with 9 days being post-conception. The blastocyst contains inner cell mass that later becomes embryonic stem cells. Because these embryonic stem cells contain the nuclei that are originally from recipient somatic cells, they contain the same genetic information as that of the recipient, and thus won't cause immune rejection when their derivatives are transplanted back into the recipient. Of course, these embryonic stem cells also harbor the same gene that causes the disease in the recipient. To prevent this from happening, a gene correction can be performed to ensure proper functionality. The embryonic stem cells are then induced to differentiate into the desired cell type, which can then be transplanted into the recipient, resolving the disease that once affected the patient. Although this process works effectively, it is time-costly, expensive, and controversial due to the origin of the embryonic stem cells from pre-implanted embryos. Thankfully, a scientist by the name of Shinya Yamanaka was able to develop a method that skipped half the steps to obtain pluripotent stem cells for medical therapies. He was able to take a somatic cell and reverse engineer it back into a pluripotent stem cell by inducing the expression of 4 critical genes that are normally expressed in embryonic stem cells. This type of pluripotent stem cell is referred to as an induced pluripotent stem cell, or iPSC. Because the induced pluripotent stem cells are directly induced from patients' own somatic cells, and thus they contain the same genetic information as that of the patient and won't cause immune rejection when their derivatives are transplanted back into the patient. Induced pluripotent stem cells consist of the exact same properties as a traditional embryonic stem cell, but without all the extra steps of somatic cell nuclear transfer and embryo culture mentioned previously. This is an incredible leap in the stem cell field because it eliminates the need to destruct a developing embryo. Most somatic cells can be induced via this process into induced pluripotent stem cells. For example, a monocyte collected from the blood can readily become an induced pluripotent stem cell under induction conditions in the culture dish. This process is referred to as reprogramming. It is a process that changes or reverts mature, specialized cells into other types of cells or back to a stem cell. The step to use somatic cell nuclear transfer is usually called cellular reprogramming, and the formation of induced pluripotent stem cells is called genetic reprogramming. Two great Nobel Prize winning scientists by the name of Shinya Yamanaka and John Gurdon helped pioneer these discoveries, with Yamanaka inducing the first induced pluripotent stem cell and Gurdon performing the first successful nuclear transfer using frog eggs. These induced pluripotent stem cells can form all different types of cells in your body, such as blood cells, skin cells, sperm cells, muscle cells, brain cells, and so on. A small sample of a person's own existing cells can be induced into induced pluripotent stem cells, which differentiate into any targeted cell type desired for transplantation. Many hospitals and institutions establish facilities to obtain induced pluripotent stem cells from patients and help the research and develop future therapy. Dr. Lawrence Darren is the director of Induced Pluripotent Stem Cell Core Facility at Harvard Stem Cell Institute. During her interview by Eric Chen, an undergraduate student at the University of Michigan, she explained the background and function of induced pluripotent stem cells, her and her team's goals, and some challenges they must overcome before applying induced pluripotent stem cell-derived cells for regeneration therapies.
We are creating the tools for the community. So we do it as a fee for service. So the investigators contact us, send us the cells, skin or blood. We make the IPS, we characterize the IPS to make sure that they are really what they're supposed to be, and we send them back to the investigator for their research. We have a broad range of samples from different diseases that we get here. Neurological disorders are probably the most part of what we do here. We had kidney diseases, we had retinal type of diseases, cardiac, so wide range of diseases. Another service that we provide at the core is gene editing. So using the CRISPR-Cas9 technology, what we can do now is we get these samples from patients with potential mutation. So we can generate IPS line, then repair the mutation, and we can give back to the investigator the IPS line with and without the mutation. So it's perfect control for their experiment. It's like isogenic line. So that's another thing that we do. The first human pluripotent stem cells were generated in 1998 by Jimmy Thompson at the University of Wisconsin. And the way he did this is it took extra embryo from IVF clinic. And these embryo are very early stage in their development, like 5 to 7 days post-fertilization. If you take this embryo and take the cells from the inner cell mass, some cells inside the embryo, put them in culture in the right condition, you can derive these pluripotent stem cells that are called human embryonic stem cells. These cells are really amazing because they have two characteristics. They can grow forever and they can turn into any cell type of the body. Now, in the process, the embryo is destroyed, and that's why it raises some ethical concerns. So the following years, in the early 2000s, a lot of researchers tried to find ways to derive human pluripotent stem cells without starting with embryo. And Shinya Yamanaka in Japan found a way in 2006. What he did is he took skin cells, and his idea was like, if we overexpress genes that are very important for pluripotent stem cells in these cells, can we reverse them or reprogram some of these into pluripotent stem cells? He started with a number of genes, 24. It worked. And then he went down to find out what were the essential genes that could turn skin cells into pluripotent stem cells. He found that 4 genes were enough: OCT4, SOX2, KD4, and CEMIC. So in 2006, this was done with mouse skin cells. In 2007, he did the same thing with human cells. So this was a big revolution for the field. The research application of this IPS line is already there. The next step, obviously, is clinical application. So can we use these iPS-derived cells for transplantation?
Functionality of induced pluripotent stem cell-derived cells is also demonstrated here by Dr. Aitor Aguirre from Michigan State University. These induced pluripotent stem cells derived cardiomyocytes beating within rhythm, just like a cardiac muscle in the heart. Another example of application is induced pluripotent stem cells forming insulin-secreting cells, formerly known as pancreatic tissue, that could eventually help treat a common disease known as diabetes. Induced pluripotent stem cells are gaining significant momentum and recognition, to the point where the NIH has taken steps towards a first U.S. clinical trial, approving the use of induced pluripotent stem cell-derived retina pigment epithelium to treat blindness due to age-related macular degeneration. With a steady increase from 2006 to now, more clinical trials are exactly what is needed for stem cells to achieve to their maximum potential. What is tricky is inducing differentiation of the pluripotent stem cells into desired cell types under optimal conditions. They need to be precise in quantity, quality, and cell type. This process can take time due to the hundreds of factors that can contribute to a robust differentiation. There are many hurdles that pluripotent stem cell therapy must overcome before it becomes a conventional method of treatment. Functionality must be 100% accurate. It must be safe from becoming cancerous, prevent immune responses, and checked by the FDA before a patient is able to use it. But it is well worth the wait and effort. Applications of pluripotent stem cells are endless. Individualized induced pluripotent stem cell-derived cells can be used for replacement therapies without immune rejection. Pluripotent stem cell differentiation can be used as a model for disease development. Pluripotent stem cell-derived specific cells from induced pluripotent stem cells can also be used to screen small molecules or drugs using induced pluripotent stem cells. Elaborating further using a great example, Drs. Eva Feldman and Kevin Chen from University of Michigan lead the current research in clinical therapy of neurodegenerative diseases such as amyotrophic lateral sclerosis and Alzheimer's disease using induced pluripotent stem cell-derived neurons. In an interview by Eric Chen, Dr. Kevin Chen discussed the potential applications of induced pluripotent stem cells in neural diseases.
For my own work, I sort of envision over the very long term establishing, say, like a pipeline. If we have a very established method to take, say, skin cells from a patient with any disorder, if we have the techniques to efficiently generate inhibitory interneurons from their cells, and once we've identified if there are any particular abnormalities within those interneurons within that disease state, and then modify those cells, transplant them back to the patient, I think that sort of facility or ability to do that on an ongoing basis, sort of like the big picture goal eventually.
Induced pluripotent stem cells are a groundbreaking discovery, an innovation that will save millions of lives, as well as improve the quality of life to those who seek it for thousands of years to come.